A screen recording for the biology instructors, lab instructors in biology 1134 for the Introduction to Statistics Week. So I just want to go through what is on the Canvas site. We have a protocol that was not there in previous semesters. This walks students through some of the things that we want them to do. I'll pull that up when the time comes. There's also the PowerPoint, which is somewhat altered from previous semesters, so you will also want to look at that. We will also go through that. Then there's some other stuff. There's a little video that I made last year about how to make a graph with error bars in Excel. There's the HHMI Data Explorer, which is what we're going to be using this semester to analyze data. And then here are some files that they'll need to do. So we're going to send them through an exercise where they draw 5 cards from a deck of cards. And these are data that I generated doing that exercise that are incorporated into the protocol up here. And then there's also a data set of the first ANOVA that they're going to do together in class. This is the P growth increment data. And then there are places for them to turn in their analysis of variance that they will do on their own and a figure that they will create. And those are the only things that you really need. You will want to download this carddraw.5.csv file onto your computer, because what you're going to do is you're going to replace the data that I have in there with data that the students actually generate in the class. And that will become more clear here in a little while. So let's just begin with what you're going to go through. So here's the Introduction to Statistics PowerPoint. And let's do it this way. So you're going to start by introducing to them about just data in general. So we talk about the different kinds of data that we'll analyze in this lab. We have data about distributions. This is what we're going to be doing when we look at Hardy-Weinberg equilibrium questions. And so the example that I have is looking at the color distribution of M&Ms, and there are 2 different sites in the United States where they produce M&Ms, but each site creates a different ratio of colors in the packages, and these are the actual published ratios from Mars Candies. And you could get a bag of M&Ms off the shelf at your grocery store and count the number of M&Ms in different colors, and you should be able to do a statistical test to ascertain whether these M&Ms that you bought came from Hackettstown, New Jersey versus Cleveland, Tennessee. And so we'll talk about those analyses next week. The other type of data that we'll analyze is data about means. We'll do this a number of times when we talk about the skull morphology of the different vertebrate groups that we're looking at. Also, when we analyze population growth rates in bacteria, we will do analyses of variance. And so we're going to be comparing more than 2 means, so they need to know what an analysis of variance is and how it works. So at this point, what you want to have them to do is you want to pause and have them open up their protocol and follow the instructions that are pretty much, I think, if I recall, just the first page of instructions. There are going to be 3 decks of cards on each tabletop: Deck A, Deck B, and Deck C. Deck A is a deck of cards that is just a regular deck of cards. Deck B and Deck C are altered so that one deck, Deck B, is, and I need to fix that, there's a mistake in this. Deck B is a low range of cards and Deck C is a high range of cards. And then once they have their data collected, they're going to bring it to you and you're going to put it into a common spreadsheet. So let's go and look at what that looks like. So I have the protocol saved on my computer. So here's what the protocol looks like. And what they're going to do is they're going to get the card deck A, and what they're going to do is they're going to spread that out on the table, and they're going to see that basically there are 4 aces, 4 twos, 4 threes, 4 fours, all the way up to 4 kings. And if we were to look at these things and treat ace as having a face value of 1, jack 11, queen 12, and king 13, you should be able to calculate what the average value of the deck of cards should be, and that value would be 7. And the other thing that you will see is that that ends up being a uniform distribution. You have exactly 4 representatives of each face value of card. You have 4 aces, 4 deuces, 4 threes, 4 fours, 4 kings, 4 queens, 4 jacks. That's a uniform distribution. So once they're done with that, have them put that deck away, and they need to break up into pairs or 2 and 3, depending on how many people are in their group. Groups of 4 would be best. 2 people work on deck B, 2 people work on deck C. And what they're going to do is they're going to shuffle up the cards. They're going to spread them out for their friend. Their friend's going to grab a card, record that value, and then put that card back in, reshuffle, grab a card, record that card, put it back in, reshuffle, until they have 5 card draws like you see right here. And then they can calculate the average value of their card draws. And they need to do that until they have drawn 50 cards from the deck so that they have 10 means. Then they're going to bring those means to you, and you are going to very quickly put them into a file that you already have open and ready for them, the carddraw.5.csv file here. And this is just a simple CSV file, and you need to save it as a CSV file. It just has what deck you're drawing, you have values for, and what the mean of 5 cards is. And then once you have that deck, or once you have all those data from all of the people in class, what you need to do is you then need to go to the HHMI Data Explorer. And what the HHMI Data Explorer is a decent way of getting students to analyze data. So once you're let me go back here, the link to Data Explorer is right here, so you should be able to just click on that and it will take you to the Data Explorer. Once you're there, and there are instructions on how to do this at the end of the PowerPoint. So once you're here, just click on Start Tool. Since I've been playing around, it's going to ask me if I want to reload stuff that I've already been working on, and I just want to reset the app. But I want to import my own data set, and then once you're here, it will ask you to choose a file, and then just go to wherever you have that file saved and upload it. And then click on OK, and it will just show you the deck and all of the values, and in this case there's 160 rows of data because you have 80 average values from deck B and 80 average values from deck C. Once you have that, you have these tabs up here, the data, the visualize, and the analyze tab. And what we can do is we can go here and we can visualize these data. And when we visualize these data, you can basically make a histogram. And in this case, the histogram is going to be the mean values. And when you do this, what you should hopefully see is 2 distributions. The low distribution is deck B, the upper distribution is deck C, and it's because this distribution just has aces up to 7s, and this deck has 7s up to kings. And what you can see from this is even with only drawing 5 cards, you begin to see something that begins to start to look like a normal distribution. And the other thing that you can see is that these 2 normal distributions don't really overlap with one another. Now, once you've shown them these data, what you can do is you can go back and call attention to things that are in their protocol where you have these distributions, and now I've made these distributions in R, which gives you a little more control over how you divide up the data. And once again, this is deck B and deck C as I simulated them. And then you have deck B and deck C with the actual population means. So up here, I've told them what I've told them what the population means are right here, because I've told them what is in Deck B and Deck C, and so we can calculate the population means and plot them there. That's not the mean of the sample means, that's the population mean. And what I want them to get across when they're doing this is this idea that you've taken a uniform distribution, you've drawn samples out of that uniform distribution, and what it gives you is a distribution that begins to approach a normal distribution. You can go back to the PowerPoint, and what you will see in the next series of slides is talk to them about the central limit theorem, this idea that when you look at a distribution of sample means, when you go back to the same population and sample it over and over and over again and save all those means, you will find that those means have a distribution that is approximately normal. And the other thing about it is that the sample means will average out to approximation of the population mean. And once again, if you go back to the protocol, you can see there's the population mean. This is the dotted lines here in figure 3 are the dotted lines that represent the average of the sample means. And then I go on and say, okay, well, the first 2 draws of 5 cards that I made out of each of these decks, the raw data are here. It produced a mean of 3 and a mean of 9.4. And we've got those plotted here, the red line here and the blue line here. And we can see that those are not necessarily very close to the population mean at all. But what you should note is that this red sample mean is very unlikely to have been generated from the same population that led to this blue distribution of sample means. This blue line here at 9.4 is very unlikely to have been drawn from the population of red cards Deck B here. And that's essentially what we're asking when we do a statistical test, is could this mean have come from this deck of cards? And the answer is no. Could this mean have come from this deck of cards? The answer is no. And so when we go back to the PowerPoint, I have a series of slides that basically talk to the students about taking this uniform distribution, applying the central limit theorem to it, and drawing out different numbers of cards, different numbers of times. So we begin here with 2 cards drawn 50 times. And what we see is that it doesn't really look terribly normal. And one of the reasons that it doesn't look terribly normal is when you draw only 2 cards out of a deck of cards, the odds of you hitting the population mean are relatively small. And so there's some explanation here that you should go through here. The red vertical line is the average of the sample means. The blue vertical lines are 1 standard deviation, and the black lines are 1.96 standard deviations. That would be the 95% confidence interval. And so this is when you get to talk to them about the empirical rule, because the empirical rule is what enables us to basically calculate 95% confidence intervals. It's what enables us to do statistics. But remember that the empirical rule only works if it's a normal distribution. Well, how do you get a more normal distribution than this? Well, you take larger sample sizes. So here's the situation when we draw 5 cards 50 times. Here's when we draw 2 cards 1,000 times. Here's when we draw 5 cards 1,000 times. And notice that the mean, the grand mean of this data set, is getting closer and closer to the expected mean of 7. And so we're pretty close there, even closer there. When we draw 5 cards 1,000 times, it's beginning to look pretty normal. Draw 10 cards 1,000 times, it's looking pretty normal. 20 cards 1,000 times, it's getting very normal. 30 cards, it's normal as can be. Now also notice that as you do this, the spread of the data are narrower and narrower as we go from 2 cards to 5 cards to 10 cards to 20 cards to 30 cards. At 30 cards, your 95% confidence interval, the range of values over which you expect to find 95% of the sample means drawn from a standard deck of cards, is pretty narrow. It's from about 5.8 up to about 8.2. And so that's not a very wide range given that the expected average is 7. And so as you increase sample size, your confidence in the mean being generated by that sample size gets substantially narrower. And this is just to contrast those two things, the two extremes. N of 2 sampled 50 times versus an N of 30 sampled 1,000 times. So at this point, there is a section of the protocol that I want them to go back to. So back to the protocol, and at this point, we want them to essentially take these first two draws that I've done, but they shouldn't do my draws, they should actually do their first two draws. The first draw from deck B and the first draw from deck C for their data that they generated. And what I want them to do is to calculate the 95% confidence intervals. So in this case, I have my data, but they should replace this with their data. And when they do that, they should get the sum of the data, the sample size of the data. They should calculate the mean, the variance, the standard deviation, the standard error of the mean, etc., etc. All of these things are explained to them in the coming pages of the protocol. So what I want you to do is just set them loose and say, read through the protocol, and it will show you how to calculate these things. As you run into stumbling blocks, ask me for help. And so I've got the formulas for it, but then I have a verbal description for it, for each of these things. This is the sample mean, and then I have it worked out for them for the data. What has happened to my PDF? Why are things disappearing on it? I have it worked out for them, and they can do the calculations themselves and come up with the answer. Then the next thing that they need to do is they need to calculate sample variance. Here is the formula. Here's a verbal description of that formula, and then here's an example of that worked out, once again, for the deck B data. Then the sample variance, standard error of the mean, and then we go through this discussion of how to calculate the confidence interval itself. And at this point, you might want to stop them, or you might want to go about this before that. But there's this explanation about what alpha means is found in both this document, and the same information is found in the PowerPoint. So this idea of what alpha is complicated for people. So alpha is basically the type 1 error rate that you set for your experiment. And the type 1 error rate is the probability that when you conclude that, for example, two means are different, they actually are not. Two sample means are different, they're not. In the example above, it would be concluding that deck B and deck C are different when in actuality they're not. And so we can control this. We set what our alpha level is as scientists, but there are some limits as to what you can do with alpha. And so there are two scenarios that hopefully will make sense to you on why you might want to relax your alpha level in the first scenario, and why you might want to make your alpha level more stringent in the second scenario. And then the idea is that they get an idea as to what alpha level is, so that they can then understand why we're using an alpha of 0.05 and why that 0.05 alpha level translates into a 95% confidence interval when we use alpha to make confidence intervals. So they should answer this question, and then they should go through the calculations, and they should use an alpha level of 0.05 with 4 degrees of freedom because they have 5 data points, and then they should calculate what the 95% confidence interval would be for deck B and deck C of their own data, not the data that are above. At this point, once everybody has gotten that done, you need to take back over and go back to the PowerPoint and reiterate some of these things. The combination of the central limit theorem combined with the empirical rule is what enables us to do statistics. And so there's a series of slides that goes through what the ANOVA does. but the goal here is for them to understand how it works, not to memorize a bunch of formulas. If you understand how it works, you don't really need a formula, and if you don't understand how it works, chances are the formula is not going to help you. But hopefully this PowerPoint will go quite a ways towards helping them understand this. So when you do an analysis of variance, it's an analysis of variance. It ultimately tells us something about means, but we don't call it an analysis of means because we call it an analysis of variance for a reason. And this is the other discussion of the type 1 error rate. You might or might not want to go through this with the whole class again, or you might want to go through this before they get to doing those calculations. But this repeats a lot of the same information that is actually in the protocol. And so at the end of all of that, we're basically asking about means and whether they're different. Go through with them what a statistical population is, and we're basically sampling from that statistical population to get our sample, and we're hoping that that sample tells us something ultimately about that statistical population. You can use the example of coots. We did this case study in lecture already, where people looked at why baby coots are colored bright orange and their parents are solid black. And you can sample coots at one marsh, but you can't sample coots that are all over the world. And so we use samples to get information about populations. And since we can't get access to the whole population, we have to use the central limit theorem and the empirical rule. And we've already talked about what those are earlier. So the ANOVA example that we're going to work with is from a textbook. It's a plant biologist who wants to know the effect of different types of water on the growth of plants. Don't ask me why they did this, but basically the researcher gave them different sugar solutions and then looked to see how much they grew. He gave them simple sugars, and then he also gave them a double sugar, and this is what the data look like, or what the experiment looks like. He gave them a 2% glucose solution, 2% fructose, 1% glucose and 1% fructose, and a 2% sucrose solution. And he also had a treatment where it was just giving them straight water, which was the control. And then these are what the data look like. So the control grew by this much, the other treatments grew by that much. So then the question is, if you just look at the data, what do you think the effect of adding sugar water is on the plants? You can see that the controls are all up in the 60s and 70s, and all the rest of these are down in the 50s and 60s for the most part. And so you might think that the controls grew better than any of the sugar solutions, but it's difficult to see what the pattern might be among the sugar solutions. But you can also look at these data in a graphical form. So I created a graph on the next slide that shows one way of visualizing the data. And you just put the growth increment along the bottom. You put the different treatments in rows essentially, and then you can plot the individual data points to see where the data points land. And we can do that for the control: 2% glucose, 2% fructose, 1% of glucose, 1% fructose, and then the sucrose. And then they're just stacked on top of one another when they have the same growth increment. And now at this point, you can then ask the question, well, what do you think is happening here? Well, it looks like the control is probably higher than sucrose. Sucrose is probably higher than these three, and these three are probably not all that different from one another. But if we want to do this in a more systematic way, we actually have to do an analysis. So we need to calculate the standard error of the mean. The larger the sample size we have, the better that estimate of the population mean is. So with 10 observations, we should expect kind of an intermediate width standard error of the mean, because 10 is bigger than 2, but it's not as many as 30. So we can take the control data, once again, have them do this, because it'll just give them more practice. The more practice they have calculating these things, the better off they're going to be. So we have these data from the control data. There are 10 values, so the sample size is 10. Once again, review how you calculate a mean. You just add all those things up, and you divide by the number that are there. this is what it looks like. The mean for the first sample is 70.1. The next step is to calculate the standard error, but to do that is to calculate the variance. So we have to look at variance. Well, what is variance? Well, if we're just looking at the control, we know that the mean is 70.1. So what the variance does is it basically looks at the distance of every point from the sample mean. And so there are 3 of those, there's 1 of those, there's 1 of those, 1 of those, 2 of those, and 1 of those. Now, if we just add all those things up, they're going to sum to 0. So what we do is we square them. That converts the negative ones to positives, and then it sums up to what the squared deviations from the mean are. So we square them up to make them positive. This is why we refer to these things as the sums of squares. Another way of saying that is it's the sum of the squared deviations of the data from the sample mean. And what that looks like, to get the variance though, we then divide that by the degrees of freedom, which in this case is 1 minus the sample size, and that will give you the variance. So the variance is also sometimes known as the mean squared, because it's the average variance of the sample. It's the average deviation from the mean. So it's kind of like the mean in that you add all them up and then you divide by sample size. In variance, you add the deviations up and you divide by 1 minus the sample size. So once again, you can go through this and they can calculate this themselves. You're going to do this calculation 3 times because there are 3 of these. You're going to do this calculation 2 times because there's 2 of those. And this will give you the sample variance, which equals 15.878. Once you have that, I've gone through here and basically kind of decomposed the equation for them. So this is just the data point. The mean is the mean, and you can see that that's the same in every one of these. And then this is just 1 minus the sample size. And so they're frightened by equations, but once you know how equations work, it shouldn't be as frightening as it was before. Standard deviation is just the square root of the variance, and then the standard error of the mean is the standard deviation divided by the square root of the sample size. And then when we go to do the 95% confidence interval of the mean, you have to know what your degrees of freedom is, which is 9, and the alpha level that you're going to be testing at, and you need to have access to a t-table. So in this case, we're going to go to the 0.05 level, and we're going to go to 9 degrees of freedom, and so the t-statistic we're going to use is 2.262. So when we do this, we're basically going to take the mean, 70.1, we're going to subtract from that mean 2.262 times 1.2601, and we're going to add that to 70.1. It gives us a lower limit of 67.24 and an upper limit of 72.95. So at this point, we can talk about how one interprets a confidence interval. One is to say there's a 95% probability that the population mean lies within that range. This is the less useful way of doing it because seldom are we trying to make statements about the population mean. Usually we're trying to make statements about sample means. And so a more productive way to do this is to say any sample mean generated from the same population as this mean has a 95% probability of falling within that range. And so that range encompasses 95% of the possible sample means that are drawn from that same population. So when we think about whether two means are different from one another, the question that we should be asking is not, do their distributions overlap? The question we should be asking is the mean of one sample included in the 95% confidence interval of another mean or not? If it's included in the other's 95% confidence interval, then that mean could have easily come from that other population. If it's not, then it couldn't have, or it's less likely to. So we can go back to these data, and what I want you to do is pause here, and each person in a group, if you have groups of 4, you've already done the calculations for the control. They need to calculate the mean and the 95% confidence intervals One person needs to do 2% glucose, another person do 2% fructose, another person do 1-to-1, and a fourth person do the 2% sucrose. And just pause, let them do those calculations, and then bring them back together once that's done. Once you've done that, here are all the answers, but they should not go to the next slide until you let them do that. And so they should be actually actively calculating these things. But here are the answers so that they can check themselves once they are done. Notice that the t-statistic is the same in all of these. Then, once we have that, we can go back to this figure and we can put in the means and their confidence intervals. And when we make vertical bars here, it's really easy to see this mean does not fall within the range of this mean. This mean does not fall within the range of this mean. These confidence intervals, though, all overlap for the most part, and as a result, we could say control is higher than everybody else. 2% sucrose is different from both the control and all of these are not really different from one another, mainly because this 95% confidence interval encompasses all of the other means that are in this experiment. And so this is how we can think about 95% confidence intervals. And this is why it's useful to plot means and 95% confidence intervals rather than other ways of summarizing data, because this way of displaying data is inferential. It tells us what the results of the analysis of variance should be. There are differences, and these differences are that these are not different from one another, but these are different from both this, and this is different from this. So, getting back to ANOVA, ANOVA is basically asking, is there enough variation among the treatment means that we can say that they're different given the variance within the treatment means? And so it's a ratio of the variation among means divided by the variation within the samples. And so we can kind of envision this as thinking of these distributions around these data, both thinking about variation among means as well as the variation within the means. If there's a lot of variation within a mean, that might obscure the variation among means like we see happening over here with these three things. So the three distributions on the left have small differences among the means relative to the amount of variation within the means, so we would say that we can't really tell them apart. The differences between sucrose and the control and everybody else are probably significant because the differences between the means are much bigger than the variation within the means. And so this is essentially what an ANOVA is going to be telling us about. We already know how to quantify sample variation within a sample, and that is basically the sample variance estimate. So you subtract the sample mean from the data points, square each of those, and add them up. This is sum of squares. It's the numerator of the sample variance equation we used earlier. In an ANOVA, we do this for every treatment, and we add them all up to give us a cumulative sums of squares within samples. To quantify among treatment variation, we don't compare means to one another, we compare means to the grand mean. So for the plant data, it would be averaging up all 50 values in the original table. Once you have the grand mean, you merely subtract that grand mean from each of the treatment means, square those differences, and multiply those square differences by the sample size. This is something that students oftentimes forget to do. And so this is once again just the sums of squares among treatments. And so they're all analogous. So here's what an ANOVA calculation would look like, and it looks like a mess, but we're going to break it down for them. What you do first is you find treatment means. The treatment means are just the averages of the treatment. In this case, there are 4 hosts, and so you average up all the data points. The grand mean is the mean of the entire data set, so it's adding up the whole data set and dividing by the total number of values that are there. And so add up all the data within a treatment, add that up across treatments, and then divide by the total sample size of the entire data set. Sums of squares within is right here. Notice how it looks very similar to the numerator. Well, it is the numerator of a variance estimate. The only thing that is different is it's got an extra summation here. So what is this equation telling us to do? It's basically telling us to subtract the mean from each data value within a treatment, square those differences, add them up within treatments, and then add those up across treatments. And that gives you sums of squares within. Sums of squares among, once again, looks very similar to a variance estimate. You're taking one mean, in this case from another mean, but this is subtracting the grand mean from each treatment mean, squaring those values. Don't forget to weight those differences by the sample size and then add them up across all of the treatments. Once you have some squares among and some squares within, you can just add those together, and that will add up to the sum squares total. So we've talked about ANOVA being used to test for differences among treatment means, but what we're really doing is we're comparing the amount of variation among means divided by the amount of variation that is within treatments. So ultimately, when we do this, we're actually going to look at this ratio of variance among means divided by the variance within means. And remember that a variance is the sums of squares divided by the degrees of freedom. So when we do this, we have the sums of squares, that's the numerator, and the denominator is the number of degrees of freedom for whatever that source of variation is. So this is a structure of an ANOVA table. We have to test the mean squared among divided by the mean squared within. This is the variance among means versus the variance within treatments. So we have treatment level variation. A is the number of treatments, so the degrees of freedom is the number of treatments divided by 1. This is the sum squares among, which we've already talked about. The mean squared is the sum squares divided by the degrees of freedom. Within a sample, it's just the mean minus the sample mean squared. That's the sums of squares. We do that for all treatments, so we need to have a degrees of freedom that is all of the treatments minus all of the sample sizes of the treatments minus 1, all added up. That ends up being the sum total of all the treatments minus the number of treatments that we have, which is A. So the mean squared is the sum of squares within divided by the degrees of freedom. And then the F statistic is the mean squared among divided by the mean squared within. And this is all laid out in here, and I forgot to animate it as I was going through and describing it, but these color codes are there to help you. Once you have this, you can look up the critical values of the F statistic in an F table. You need to know the numerator and denominator degrees of freedom, but the numerator and denominator degrees of freedom come from this ratio. What's the numerator degrees of freedom? It's the degrees of freedom in the numerator, the among. What's the denominator degrees of freedom? It's the degrees of freedom for the denominator, which is within. So your example has 4 numerator degrees of freedom and 45 denominator degrees of freedom. And because this table starts jumping in bigger steps as you get bigger and bigger sample sizes, it is of no use to you. But you can go to a statistical calculator. You can plug in the degrees of freedom and the F statistic that you get, and you can find out what the p-value is for your test. So the next thing that they have to do is analyze the growth data using the analysis of variance. They know how to get the sums of squares because they can just multiply the sums of squares times the number of degrees of freedom, and that will, they can take the variance, multiply it times the degrees of freedom, and that will return the sums of squares. It's just a mathematical manipulation of the sample variance, equation. So their next task is cut them loose on this, and they need to work together to get the ANOVA table done for the P data. And this will take most of the rest of their time. And once again, you should be circulating around the room doing, helping them with this as they need help. And then they have an ANOVA table down here that is structured for them. They need to fill that in. Once they've done that as a group, their homework is to basically do this with a different data set by themselves, not as a group. So this is the result for this. Now, what they can do is they can also take these data and they can put them into the Data Explorer. So they can download the PEAS data set, they can open that up and they can see how it's structured. Then they can go to Biointeractive, they can follow these instructions, and as they follow these instructions, they can basically check to see if this analysis that they just completed in class matches what is in the Data Explorer. So let me go back to Data Explorer and show you what that looks like. So what you can do is just reset the app, reset without saving. Now we're going to import our data, but in this case we're going to import the peas.csv data file, which is actually on Canvas, so you can download it before and play around with it. Once you have it uploaded, you will see that it's got the control, 2% glucose, 2% fructose, etc., etc. And now we can go to Analyze, and when we go up here, we can add a new analysis, and we want to compare means of 3 or more samples, and we want to do a one-way ANOVA assuming equal variances. And when you click on that, it's going to give you down here in the lower right-hand corner, it is going to give you the result. And so I say it's, it won't because we haven't told it what the variables are. The dependent variable is growth increment, and the grouping variable is treatment. And once you do that, it's going to give you the means, the standard deviations, and standard errors of everything, so they can check their calculations of those things. But then it's going to give you the information that you would normally see in an ANOVA table. It gives you the sums of squares among groups, the degrees of freedom among groups, and the mean sums of squares for among group variation. Within group, once again, sums of squares, the degrees of freedom, and the mean square. 269.33 divided by 5.5 is 49.368, and that should be what we saw over in our result in our PowerPoint, 49.368. And so you can they can check their math and make sure that they did everything correctly by importing their data into here. Now, once they've done that, if they want, they can also go over here to visualize, and they can add a new plot, and they can do a box plot, and their box plot should once again have treatments on the x-axis, and the thing that you're wanting to plot is the growth increment, and they can create a box plot. And sure enough, it looks like control is higher than 2% sucrose, and 2% sucrose is higher than all the rest of these, and all the rest of these aren't really very different from one another. And so this is not a mean and 95% confidence interval is a confidence interval plot. This is actually a box plot, but once again, there is a video online that shows them how to make means and 95% confidence interval plots if they want to do that. So now that they have checked that, they can then go to the next slide, and on the next slide is the tick data. It looks at hosts. In this case, the sample sizes for each host is somewhat different, but once again, in the end, they need to calculate the summary statistics. Once they've calculated the summary statistics, they need to calculate the sums of squares among, within, the degrees of freedom, mean squared, F and P. They need to turn essentially this table in to you, and they need to create a meaningful graphic of these data as well. And so in the room, you're going to have blank PEAS dataset tables. You're going to have blank tick dataset tables. The tick dataset ANOVA is what they need to turn in, and they need to turn this whole thing in so that you can see that they have done the work, and they need to together work on a meaningful graphic of the data. And so when you go back to Canvas, once again, why can I not get back to Canvas? Oh, there's Canvas right there. So the analysis of variance is an individual assignment, so you should have as many of those as you have students in the class. And the data figure is a group assignment, so they need to work on constructing that together. And so when they finish doing their tick analysis by themselves, they should get together and compare results with one another, or they should maybe even put those things into the Data Explorer and get it to calculate the ANOVA for them. And that will also give them the means and the things that they need to calculate a confidence interval as well. And so they need to turn in that as a group, and they need to make sure that everybody's names are on it. So that seems like a lot, but once again, have them do things, pause, introduce them to the next thing, have them do things, pause, introduce them to the next thing. So it's a series of you saying things, them working, then you saying things, then them working, you saying things, and them working. I think that's it. So let me know if you have any questions between now and next week. I'm going away for the weekend, but I'll be back Sunday. So if you have any questions that you want to meet and talk about on Monday, feel free to do that. I'm free all day Monday.